This last part of the lecture that we didn't quite get to on Thursday in class. And I promised a couple of times or in the last week in class that I would get the last bit that I wanted to say done before spring break, so that when we get back on Tuesday, there can be a day full of questions. There might be new material coming up as part of answers to the questions, either in class or that somebody has emailed me. But other than that, we won't have to have new material 2 days before the exam on Thursday, and the 26th. And so that's what this is about, is just finishing up that last piece of what I definitely want to talk about, chapter 19 and material that'll be on our exam. And so on Tuesday, I'll answer questions if there's time. I'll just start talking about new stuff that won't be on the exam. Excuse me. Yeah, so this is where we're at. So in previous editions, the book had a just well-organized list of characteristics that it wanted to talk through that are kind of common features of cancer cells. And so the first two we talked about in class, actually the first five, but to look through these. So these are really, they go hand in hand, right? So uncontrolled proliferation, because they have reduced dependence on growth factors because they have mutations in oncogenes like RAS. So they're just going to keep growing like that. And then these two were from those dishes of sterile tissue culture cells. And when you culture normal cells in a dish, they're mortal. So they come out of a mortal person. And the individual cells in a dish, it's kind of interesting to see, they're all still themselves mortal. Cancer, on the other hand, is interestingly immortal. And so it's got things that have happened to it that have immortalized the cells. And then cancer just keeps growing in a dish. So normal cells have a kind of growth limit where when they make contact with other cells and use up the surface of the growing material in the dish, they stop growing. Cancer cells just keep growing. And that in the body, it seems like that's part of what contributes to the spreading of cancer, the metastasis of cancer. Cancer cells in a tumor start to use up all the nutrients and all the oxygen, and gas exchange available. And so if it spreads out, it'll just be a selective advantage, and that's what happened. And you can see that in a dish where the cancer cells are just less willing to hold on to other cells and hold on to the extracellular matrix. That's the mesh that's holding them in place. So then we talked about secretion of proteases. Here's an image of that. Cancer cells spreading, and they can spread partly because they're chewing up. They're releasing molecules to degrade this mesh, this extracellular matrix that holds them in place. And also they can break down the proteins that are holding cells together. So the cells are holding hands, essentially, and the cancer cells can chew their way through that between cells and spread. So this is just a solid tumor getting into the bloodstream. So that's that idea of releasing proteases. Then the next one is related to what I just said, and that is, this cancer, as it gets a bigger tumor, it needs to recruit new blood supply. And so new blood vessels have to form, and that's called angiogenesis, and that's the formation of new blood vessels. And so new blood vessels and cancer tumors are big tissues, big solid tumors, so that need to recruit blood supply, both for bringing nutrients and oxygen and taking away carbon dioxide and waste, and of course other molecules as well. But that's the center of it. And in general, new blood vessels grow in response to anoxia. Anoxia is just a lack of oxygen in an area, and they'll stimulate new blood vessel growth. And so that's what the center of a tumor is like. It is a necrotic, that means dying cells, difficult environment, because it's hard for the the tumor to recruit new blood vessels. And so for years and years, I think I have a picture of that. Yeah, so just new blood vessel growth. And for years and years, I guess that is both the spreading of the tumor and then at a new location releasing molecules to increase angiogenesis. And so angiogenesis, and years ago, maybe 40 years ago, a scientist hypothesized that if we could inhibit new blood vessel growth, we could inhibit tumor growth. And so there are actually several cancer drugs that do that. And it is a pretty effective strategy to reduce the rate of tumor growth, is to limit that. Because in an adult body, there is very little healthy new blood vessel development going on. And so, it's okay to inhibit that and in the whole body, and that can help slow down the cancer. So that gets on this list because it was a big idea in the lifetime of our author, and it turned out to be an effective strategy. Here's one I'm going to describe because it is sensible for us, but different than what we're used to. And what it's showing is this. So this is that stem cell in the bone marrow that we've talked about, where it's making lots of new cells every day of our lives. So this is the new blood cells getting produced in the bone marrow. So that stem cell's dividing, and it divides like this, where it divides equally and makes more stem cells like that. So it can divide like that, and it can divide asymmetrically like this, so it can keep making stem cells, but it also makes what's shown here, these precursor cells. And so these precursor cells are precursor blood cells, and they're showing that those start to divide, and they have to make all the different types of blood cells, like red blood cells and white blood cells and T cells and B cells of the white blood cells group and so forth. And so there's lots and lots of cell division, and as they divide, there's a point where they start to undergo differentiation. And we learned about differentiation. So we're in our muscle videos at the start of our course. So this is the same thing as that. Instead of making muscles, we make blood cells. Sorry, keep saying that wrong. And so what's happening here is that we learned when the muscle differentiates, it stops dividing. And that's a general truth in differentiation, that differentiated cells tend to stop dividing, really likely, like that, really kind of typically. And so that's what's happening here. But what happens here is that there is a mutation, not in an oncogene, and not in a tumor suppressor, but it's just a different category of a gene that could also mutate and cause uncontrolled growth. And that is, there's a mutation that blocks differentiation. So the cells fail just to fully differentiate, and that because of that, they just keep dividing. So they're at this level, and they're just going to keep dividing instead of stopping as differentiated cells. So then just bring it up one time, that not every story in cancer is a story of an oncogene or a tumor suppressor, and sometimes there are other situations like this, where in this particular example, the cells will just keep dividing because they just can't differentiate and stop like that. They can't finish the job of development. Yep, like that. So then another one, and this kind of loops back up to here, fits into here very well, where we already know about good examples of that. And so here's an example of that, where there can be an extracellular signal that leads to the activation of the caspases and programmed cell death through that cell signaling. And so the cells should die under some normal conditions, and they just don't die because they've had mutations in that pathway. And so our example of that is that BCL2 antibody gene connection in that translocation in low-grade non-Hodgkin's lymphoma. Okay, like that. Yep. All right, great. So just a couple more things. Here's an example that's in our book. I just want to point out that this is the problem that we talked about with the first exam with the woman with skin cancer, where she has the PDGF skin collagen gene translocation. And so it's an unusual example. It's called autocrine. We have endocrine and paracrine from endocrinology, and this is autocrine, and it just means self-stimulation. And so that's all that's happening here, is that they're just showing the cell dividing. They don't even need to show that. They can just show one picture because the cells are all doing the same thing. And that is they're making a growth factor that they have a receptor for. So there could be coming out of there, that could be PDGF being made because of the translocation in these skin cells. And it just binds to the PDGF receptor and activates the pathway. And the RAS, RAF, MEC, ERK, ELK, and so forth, MIC, everything else is healthy. A very healthy wound response is being stimulated in this woman. The cells are going through the cell cycle, thinking they're helping make more skin cells quicker so we can heal this wound. But there's no wound. It's just that there's been this translocation, and because of genomic equivalence, the skin cells have the PDGF gene, and if they make the mistake of translocating it, with the collagen gene so it gets expressed, well, we can get this autocrine stimulation. So I just wanted to point it out because we have this really good example of that. It's a very unusual thing in in biology, because what is the point of having a cell stimulate itself? But there are some good examples of it in normal biology, and we just won't get into them here because it comes up because of this cancer story. Yep. And so I just want to show you this, and I think this is my last slide. Yep. And I just want to show you this, and I've said it out loud, but to add in the assignment about the CAR T cells therapy, and I say it there too. So for major treatments of cancer, we should know that there's radiation. So try to stop the tumor by exposing it to radiation like X-rays and other radiation sources to mutate the cancer cells and kill them. And then surgery. Let's try to remove this tumor. Then, and I should just mention that this is for solid tumors and those cancers of the blood, the leukemias, lymphomas, myelomas, those are not solid tumors. And so, surgery is not a possibility for those generally. So then there's chemotherapy. This would be any kind of drug. And so we have a good example of that. It's tamoxifen. And then we learned some about immunotherapy, and we have a good example of that. And in the homework video, I talk about more examples of immunotherapy, but that's certainly one, and there are others that we are also learning about, because CAR T cell therapy is also an example of immunotherapy, but we'd like to put it in this new category that's a very kind of a new philosophy and strategy, and that is let's try to manipulate cells. And in the case of CAR T cell therapy, we make we use a transgene to make a cell transgenic. So that's a dramatic example of cellular therapy, right? We're really going to modify that cell. Other types of cell therapy would be just that we somehow favor a certain cell. So that's talked about in the homework video of adaptive immunotherapy. And so anytime we harness a cell to get the job done is a new kind of style. And that's it. So this is all I want to talk about prior to the first exam. However, again, some more material could come up on Tuesday and, in emails or something, Brightspace announcements with regard to questions students have. So let me know about questions as we're getting ready for the second exam. Thanks a lot.